Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is June 16, 2025. This video is on a review article published in the New England Journal of Medicine on June 4, 2025, titled The Fetal to Adult Hemoglobin Switch, Mechanism and Therapy, authored by Dr. Stuart Orkin, MD. Switching gears, today we are unlocking the secrets of hemoglobin. Not just the protein that carries oxygen, but a molecular switchboard that may just hold the key to curing sickle cell disease and thalassemia. Let's dive into this remarkable journey from fetal blood to frontier therapies. The biological switch. Before birth, our red blood cells run on fetal hemoglobin or hemoglobin F. But as we take our first breath, a molecular baton is passed to adult hemoglobin, HbA. This switch is vital, and in sickle cell disease or beta-thalassemia, it can turn deadly. Yet, increased fetal hemoglobin levels have long been known to offer protection, a fact that first observed in infants symptom-free until fetal hemoglobin faded. BCL11A, the master repressor, it is a gene that silences fetal hemoglobin. Identified via genome-wide studies, it binds and represses gamma globin. Gene editing targeting BCL11A's enhancer, particularly the GATA1 site, reactivates fetal hemoglobin and restores hope for patients with hemoglobinopathies. The CRISPR revolution, with exa-cel CRISPR-Cas9 being used to deactivate BCL11A enhancer. Over 90% of patients in trials were freed from transfusions or crises. But this high-tech therapy demands sophisticated infrastructure far beyond the reach of most affected regions. Beyond the lab, global access, the future lies in small molecules targeting BCL11A or co-repressors like LRF. From PROTACs to epigenetic agents, mechanism-based reactivation of fetal hemoglobin could democratize therapy. PROTAC stands for proteolysis targeting chimera, a novel drug modality that induces selective protein degradation rather than merely inhibiting function. It's revolutionizing drug discovery for targets previously deemed undruggable, including transcription factors like BCL11A. All roads may lead to BCL11A, but the route must be many and accessible. Today's episode is about more than science, it's about changing destiny. With every edited gene, we inch closer to cures. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by Master MedFacts at mastermedfacts.com.